So today I'll be talking about electron bifurcating enzyme. So I'm from the SIBYLS group at ALS. This is in collaboration with the Mike Adams group at University of Georgia. Okay, so yeah, basically basics of electron bifurcating enzymes are that it's a 2-electron reaction. Basically, this enzyme accepts 2 electrons. One of those electrons end up going to this low potential acceptor. So this is more reductive. And then the other electron goes to this high potential acceptor. So this is less reductive. So basically by coupling these 2 pathways together, it basically, the net is overall increase in potential, meaning it drives the reaction forward. And so that's how it can able to get these more reductive acceptors, which are important in anaerobic micrometabolism for like reducing their carbon source or reducing nitrogen and other things. And so that's kind of why people want to look at this, is because like producing like hydrogen gas or fixing nitrogen. So I am looking at, in this case, this electrotransfer flavoprotein ABCX. And so basically, there's still quite a bit of like uncertainty of like the mechanism of how the electrons are being transferred. This is like currently the most model, like newest model that's come out. There's been several iterations where it's quite different in what's doing what. But the basic gist of the reaction is NADH reduces ferredoxin, which is used to do other more like things because ferredoxin is highly reductive, and then also it reduces metaquinone, which it can use to drive a proton gradient and recover the energy by making ATP from that proton gradient. So okay, so going into the data, so collaborators sent us EtfAB from Pyrrhococcus aerophyllum. So basically what we wanted to do is see if there's conformational changes under different conditions. So basically you have EtfAB by itself, no additives, EtfAB with dithionite, which is a very strong reductant. So that should basically make sure it's like completely reduced. I have this condition where I have dithionite and NADH. So basically make sure it's completely reduced, but also have the NADH so it can bind and see if just the NADH by itself was doing something regardless of the reduction state. And then I also did a NADH pretreatment where the sample was pretreated with NADH, but since this was SAXS, there was no NADH in the running buffer. And then a condition where there was actually NADH in the running buffer. As you can see, here's the P of R function, which is the most probably dramatic thing to look at. You can see that in the case of NADH, it's getting larger. So something is like kind of probably popping off and extending out, versus all these other conditions are pretty much the same. So I've done some modeling with this. So this is the no additive case. And so basically I made Roberta models and also compared to like other homologous enzymes where there's crystal structures to rearrange the 2 subunits. So I did some modeling with this and got chi-squared of 1.26. So it looks like it's a pretty good fit for the no to additive, and it's kind of what you expect based off the literature of looking at similar enzymes where structures are solved. So that's great. But then I go to the NADH case where it seems to be elongating. So I did some modeling with this. Basically what I got is I've modeled some different states. Right here, I'm actually just showing the 2-state for simplicity. There's basically 2, they're overlaid, like one's a slightly lighter version, the other's a darker version. But basically what I got is that this domain, in the presence of NADH, some proportion of the enzyme is having this domain pop off and be extended out from the rest. And you can see like what the model is saying, what percentages of it. So like 59% in the 4-state model is this like the compact state, and then the rest of it are these varying other more extended cases where something is popping up. So that's what I've done so far. And so this is just looking at EtfAB and in this particular species. So collaborators are also looking at one from Thermatoga maritima. And so in this case, this is actually HC-SAXS. So there might be some issue, like looking at the data with some aggregation might be causing a little issues, but so yeah, I basically did much of what I did before. No additive, having dithionite, NADH. And in this case, I did NAD plus just to see if the NAD, like having the NAD in there, like binding it was doing something, or if you needed NADH to actually reduce the protein, and that was actually what's doing it. So this is actually a little different from what I got, the data I got before in the SAXS case with the other species. No additive is the smallest, but it seems that like dithionite and the NADA plus also are causing it to be somewhat larger, but then also, but the NADH is creating it to be the largest. And so in this case, they also are able to get the EtfABCX full complex. And so again, you actually see pretty similar trend in that the no additive is the smallest. With the dithionite NAD plus, you see it's slightly bigger, and then the NADH is showing it to be the largest. And then if you actually look at these curves, you can see that there is a slight difference in the shape between the dithionite and the no added and dithionite case versus the NADH and the NAD plus case, where this is slightly curved more than these two. So it looks like NAD or NADH by itself might be doing something in the conformation. So I've done some modeling with this. So in this particular protein, just this year, they've actually managed to publish a cryo-EM structure of it. And so basically I'm trying to use that as like the base to do some modeling. So comparing my SAXS data to their cryo-EM structure, you can see the fit is pretty bad. So basically their cryo-EM structure was a super dimer. So if you compare the data to just what would be a monomer, not a great fit. If you look at the full super dimer, it's also not a great fit. And you look at the PRR function, it doesn't fit well. So basically that means something else is going on with this protein in regards to the solution state SAXS data. So I have tried doing some more modeling with this. So basically I tried computing a shape, and this kind of is like one of the better shapes that that looks like representative of what it is. And so basically I'm still in the process of doing this, but I've added in like other, this is basically trimer and a half, and this case is like a trimer. And so if you look at the comparison, both these are the SAXS curve and the PFR, it looks like it fits better. So it basically looks like their cryo-EM structure probably is pretty correct, but either there's something else like it's sticking off, and that could be like a stoichiometry issue, and that like all the subunits might not be in correct stoichiometry in the sample, or it could be that there's more conformational exchange in this protein, and it could be possible that what they saw in the cryo-EM was this structure, but it could be possible that this is actually it like flips out or something, and like what exactly is going on is not fully certain at this point. I probably actually want to do SEC-SAXS to see if we can actually separate if there are different conformations that are running differently. So yeah, so just to keep this short, this is what I have. Acknowledgement to my other lab members, Mike Adams group, and then funding.
Yeah. And we should mention here that Jan did all of these under anaerobic conditions, so no oxygen at all, or the ones that you
So it's important. Yeah, so these experiments are a little more difficult because it is important to keep oxygen out. So you can actually maintain a reduced state and it's not like oxidizing over time and messing with your results.
So if we do have people out there who are interested in doing anaerobic experiments, Jan is the guy to talk to. Not only has he got the infrastructure to work anaerobically up at the lab, but he's also done a lot of the spectrophotometry that goes along with characterizing and ensuring that it's in the state you think it's in, which is also really important. Yeah, for those of you considering anaerobic projects, they're a next level project just because of the anaerobic stuff.